It is well known that exhaled breath VOCs can easily reflect certain metabolic pathways. On the other hand, external agents such as diet and lifestyle habits, for example physical activity or alcohol and tobacco consumption, environmental pollution, etc., can affect levels of specific breath VOCs. Some of these volatile compounds are acetone, ethanol, isoprene, and N-pentan. Therefore, there is potential to use these exhaled breath VOCs as biomarkers for the improvement of quality of life and general health. For accurate diagnostics, an individual approach is required. In order to enable wide individual screening, utilization of affordable solutions for on-site analysis is needed. These screening results would complement conventional diagnostic techniques for more efficient nutritional treatments. Our contribution to this goal is the introduction of a portable mass spectrometry sensor for determination of breath VOCs which are related to food metabolism and lifestyle. This instrument is developed and validated by Bioscience Institute team within European Horizon 2020 project Protein. In this study, exhaled breath samples were collected in 1-liter toddler bags from 44 participants, professional athletes in Greece and Portugal. Each participant provided 2 exhaled breath samples, one before the meal, after 12 hours of restraint from food, and one 120 minutes after the meal. The nutritional information for provided meals are shown in tables 1 and 2. The participants were divided in 2 groups. 24 participants from Greece were in steady state during the period between 2 samplings, while 20 participants from Portugal performed their regular training between samplings. Along with the exhaled breath samples, all participants provided some information on their lifestyle habits via questionnaire. This information is summarized in paragraph 1. Concentration levels for selected breath acetone, ethanol, isoprene, and N-pentan were determined in samples before the meal and after the meal using a portable membrane inlet mass spectrometer showed in Figure 1. It is equipped with electron impact ion source and quadruple mass analyzer. It provides a mass scan range up to 300 Daltons, and its dimensions, weight, and low power consumption enables portability. Obtained breath acetone, ethanol, isoprene, and N-pentan levels in samples before and after the meal for professional athletes are presented in bar graph 2. These were in the expected concentration ranges according to the available literature data. Food impact on selected breath VOCs was expressed via comparison factors, which represent calculated ratios between VOC level determined in sample 120 minutes after the meal and level in sample before the meal for the same participant. If the calculated value is above 1, the VOC level increases after the meal, and if that value is below 1, then it has been decreased upon meal. The paragraph 3 presents comparison factors for 2 groups of professional athletes. In Table 3, percentage of significantly altered VOC levels upon meal consumption are shown. An increase or decrease in exhaled breath VOC level greater than 10% was considered as a significant change. Taking that in mind, it can be noticed that significant change upon meal consumption was observed in about 75 to 80% of athletes who were in the steady state between 2 samplings for acetone and ethanol, 33% for isoprene, and 88% for N-pentan. Moreover, a significant change upon meal consumption was observed in 55% of athletes who were physically active between 2 samplings for acetone and ethanol, 70% for isoprene, and 50% for N-pentan. Comparing obtained exhaled breath VOCs concentrations against data collected via questionnaire, some statistically significant correlations were revealed. Impact of participants' category, gender, working environment, and alcohol consumption were observed for breath ethanol and isoprene levels. Moreover, acetone level was affected by participants' category and gender, while N-pentan level was different among participants who consume alcohol and those who do not. Future work could expand the number of breath VOCs and involve more participants in order to get more reliable conclusions and to contribute to the breath research field. The authors would like to thank the project partners who recruited the participants. This study is funded by European Commission Project Protein, Personalized Nutrition for Healthy Living, and supported by Antares Project, both from Horizon 2020 Framework and Serbian Ministry of Science.
Validation methane and hydrogen portable breath analyzer, air 2 through calibration gases. Previous studies used exhaled gas from volunteers as a test gas for validating the first version of air. This study tests the next generation device. A mixture of certified calibration gas is used as the reference gas. The study addresses shortcomings of the initial approach and use pretest technical gas mixtures for validation. To obtain various concentration of the calibration gas, a setup shown in the block diagram was used. It included two cylinders with certified calibration hydrogen and methane, a humidity generator and dry air generator. Programmable flow controllers were used to control the concentration. The error of this calibration system is no more than 5% in the range from 10 to 100 ppm. For convenience of conducting the experiment, a special stand was assembled, allowing for the control of flow through the AR2 devices and their simultaneous calibration. This slide shows the dependencies of the score on the concentration of hydrogen and mixture in the absence of methane. In both humid and dry air, it's evident that in humid air the characteristic cease to be linear. The score in humid air is higher than the score in the dry air. None of devices indicates the presence of fermentation in the methane concentration of 11 ppm, and only two devices were sensitive to methane at concentration of 55 and 110 ppm. There is an evident higher variability in the score values from experiment to experiment for all devices. Additionally, AR2 signals the presence of hydrogen in the mixture, even though hydrogen is absent in the calibration mixture. Overall, the fermentation level score is more sensitive to the concentration of hydrogen than to methane. For instance, at a hydrogen concentration 27 ppm and methane concentration 55 ppm, the device shows a zero level of methane and high hydrogen score. According to the AR2 device reading hydrogen affects the fermentation level more than methane. However, research suggests that methane actually has a greater impact on the fermentation level. The AR2 device underestimates the concentration of methane, especially at low values in the target gas. In our opinion, the issue lies with the methane sensor used, whose sensitivity is guaranteed at methane concentration above 100 ppm.
Hello, I'm going to introduce our poster, Development and Validation of a Portable Device for At-Home Hydrogen and Methane Breath Testing. Hydrogen and methane breath tests, also known as HMBTs, can be used in clinic to diagnose small intestinal bacterial overgrowth and carbohydrate malabsorption due to the connection between the levels of these gases in the breath and the activity of the gut microbiome. Longitudinal measurement of hydrogen and methane also allows for monitoring treatment response and early detection in disease recurrence. For this purpose, OMED Health has developed a portable hydrogen and methane breath analyzer device. In this study, we completed several validation tests to measure the accuracy of this analyzer device. We developed a gas concentration predictive model simulating human breath and allowing us to measure the concentration of hydrogen and methane within a mix of gases. We also benchmarked the OMED breath analyzer device to an in-clinic hydrogen and methane analyzer, the Gastrogenius breath monitor, using the breath of human volunteers. 941 synthetic breath exposures were included in this study, and the desired concentrations of hydrogen and methane were generated by calculating the ratios required of constituent supply gases in a synthetic breath gas flow. The results found that the sensors within the OMED device and an analytical model utilized as part of the device can accurately assess and report hydrogen and methane levels. The OMED device was then compared to a clinically validated HMBT instrument. A total of 243 breath samples were collected from 54 participants. Each participant gave 2 samples into the HMBT, and the mean was calculated. In between both samples, the participants gave an extra sample into the OMED device. Treating the results of the average of the HMBT as a truth, there was a comparable calculated error for hydrogen and methane between HMBT and the OMED device. In a preliminary study assessing the diagnostic performance of the OMED device, this has shown a high degree of concordance with the HMBT tests, achieving more than 90% accuracy in both SIBO and IMO diagnoses. In conclusion, this data suggests that the OMED device is highly accurate with comparable performance to an in-clinic HMBT. The added portability for convenience and the possibility of taking regular at-home measurements can provide significant benefits for users by providing longitudinal data that it is immediately available for their healthcare provider. Thank you for joining me and hearing more about our poster today. I hope you enjoy the rest of the conference.
Dear colleagues, I am pleased to share the results of our study on behalf of the team from the School of Medicine and from the Institute of General Physics. The main idea of our work was to analyze the kinetics of the release of hydrogen and methane produced by the gut microbiota by using tests with water saturated with hydrogen and methane, because the final pathway for clearance is the same for both cases when these gases absorbed from the microbiota or when they're absorbed from the drank water. And it passes through the liver and goes via lungs and exhaled with the air. So for this, we used the test with water. And here we describe how we prepare this water saturated with hydrogen and methane under 1 atmosphere. To know exactly concentration of these gases in the water, we used quintron gas chromatograph and analyze concentration in overhead over such water. So after that, the volunteer drank this water, and we recorded the curves, the concentration of these gases in exhaled air. So what was the result? We showed that the whole release, the whole clearance takes around 40-45 minutes. And we observed differences in dynamics of release of methane and hydrogen. So as well as we know exactly the concentrations of the gases in the drink water, and we know exactly the concentration in exhaled air, and we know the minute volume of exhaled air. So we know exactly the number of molecules of hydrogen and methane that was consumed by the volunteer and the concentration, the whole contact of molecules that was exhaled. So this allows us to calculate the percentage that was consumed by the body, and we expect that mainly by the liver tissue. And it was shown here. You see here curves of the real experiments. A young woman, 24 years old, drank this test water with hydrogen and methane, and we saw that 64% is consumed of the hydrogen and just 57% of methane. So usually much less methane is released with exhaled air. It could depend on the differences in saturation of liver tissue or in differences in permeability of the intestinal wall. So finally, we make a conclusion that using this test, we can better understand the kinetics of the gases generated by the microbiota. And the second one, we can add lactulose or carbamide. And in this case, during one run, we can get much more information concerning the kinetics of these gases. And like in a regular breath test, we can understand better the metabolic activity of the gut microbiota. Thank you very much.